Thanks very much. Going last at the coffin corner of a meeting when people are tired and some people aren't here is not easy. I'm going to give what will be maybe a kind of rumination or a philosophical overview of some of the things that have been said during the meeting. And since I didn't know exactly what was going to be said, I'll be adjusting what I say about my own slides just because to try to adapt. I think we are faced with some epistemological challenges that derive from our use of what I would say are Enlightenment era, 400-year-old epistemological criteria for science, largely developed for physics. Among the criteria that we use all the time, and many of which have been mentioned here, are replicability, but life, because it's an evolutionary history, doesn't guarantee repeatability, and fitting of survey data, for example, is not the same as prediction unless you have a theoretical basis for doing that. Falsification you hear talked about all the time, especially by geneticists. Often, I think, I've never read any philosophy of science, but in the absence of a guarantee of replicability, falsification is not a good inferential criterion. You can falsify a hypothesis because you have a crappy experiment, but you can also falsify a hypothesis because another sample may not have the same characteristics because we're dealing with life. And life doesn't always replicate because that's what the evolutionary process does. Similarly, verification, usually you want to specify criteria for verifiability of an assertion, but verifiability is a kind of weak criterion because you can't always say how you expect a sample design or an experiment to turn out, for the very reason that life is a complex result of a complex history. Predictability is often said to be the coin of the realm or the gold standard, but even with perfect theory, which we certainly don't have in biology, measurement errors and other things can vitiate predictive power, and things like chaos can mean that even a small amount of error can lead to uncontrolled errors in prediction without our being wrong in any serious way. And finally, I also hear parsimony invoked all the time by people about their explanations. And parsimony is, life does not follow a geodesic path. Parsimony is not a guarantee. It's not even easy to define what parsimony would even mean. And life is not parsimonious. So it's easy term to use. It sounds like you know what you're talking about, but it's a lot harder to make sense of it. So in science, there are things like Platonic idealism, by which the Pythagorean theorem holds for every single triangle, along with the angles summing to 180 degrees, in this case, right triangles, except that we know that there's no such thing as a right triangle, because every triangle is imperfect. And yet, somehow, even on the farthest galaxy, we believe that the Pythagorean theorem would hold. This is a kind of Platonic idealism, that there is some kind of reality to things that we don't actually see except in imperfect manifestations. There are also some criteria for externally derived ways to apply the concept of natural law to the real world. Over here I have Newton's law of, Newton's law of gravitation, which is a deterministic law. If you know the masses of two objects and where they are, you can tell what the attraction will be between them, and you can do it essentially perfectly except for measurement error. In the 19th century, statistical regularity was recognized in things like the ideal gas law, where you know that you can't follow every individual atom, but you treat each atom as a billiard ball, and you say that statistically we can at least predict some aspects like pressure or temperature of a volume of gas. These kinds of things lead to externally derived theory that we then apply to new data. And I would argue that in biology, to a great extent, often where it counts for this kind of a meeting, all we have are very inadequate generic theory, and we have to use internal comparisons. Internal comparisons such as in this genome-wide map. If you don't know what a genome-wide map is, this just goes along all of the genome. You look at all sorts of markers, variants across the genome, and you decide how statistically unlikely are cases to have more copies of this per capita than controls, and you decide by some subjective and arbitrary cutoff called a p-value or whatever, or likelihood ratio scores or something like that. You decide that this and this point count. These ones don't count, and you're supposed to follow your statistical assumptions, but if they don't count but they're close, you say it's suggestive and you write a grant to follow it up, instead of saying, I didn't find what I thought and therefore I'm going to go do something else. And then down here, these we say, nah, these don't matter because they don't generate statistical significance. Well, I'll try to talk about that a little later on, about what the implications of ignoring all this are. And what I would say in general is that life is a kind of ad hoc history, which we call evolution. It's a context-specific and I'll use the word Boolean or combinatorial set of relationships among non-homogeneous polymeric elements, genes or DNA sequence, regulatory regions, interaction networks. And that's what we're trying to deal with. And a lot of the issues that have come up here, either explicitly or implicitly, reflect this. Often when you have this kind of a setting, a situation, what counts is the collective location or arrangement of these various elements, not their presence or absence. And each trait, each gene, and each population is inherently different. That's what evolution basically means. And life's causal architecture is statistical, not specifically predictable, I think, in principle, just by the way these things act and interact. Now, I would say we should ask ourselves how often we're asking well-posed questions. And this just shows a schematic of a structure of a particular gene, ApoE, that's involved in some human diseases. And what is a well-posed question? Well, here is DNA sequence. It's the ApoE gene in humans. DNA by itself is a random sequence. It doesn't do anything except decay unless it cooperates or interacts with other molecules. So I would ask you this question, which I could probably stop right here and say, this is the most profound kind of question that we should be asking, and we should be asking it seriously. If this nucleotide, which is a T, is changed to a G, does it have an allelic effect? That's the simplest, stupidest question you can ask in genetics, and it's one of the very hardest to answer. Does it have an effect? How would we know? How would we know if even this simple change, one nucleotide to another, has an effect? I don't know if you can see why it's such a complex question, but the answer is, in my opinion, that the effect is strictly dependent on the specific genomic context. That is, on the sequence elsewhere in the genome, which we rarely know except incompletely. And as a matter of fact, everybody's elsewhere genome is different. We all know this, but I think we don't take it quite seriously enough. And I'm not going to give you answers because I don't know any answers. But you can say, here's this gene, ApoE, and you can say this nucleotide has been associated, change here at this position along the gene has been associated with Alzheimer's susceptibility and various things. And people would say this site affects lipid levels, or this site affects Alzheimer's disease. Well, how do you know that? For example, maybe it's this site. Or maybe it's those sites. Or some combination. Or what combination? We each have two copies of the gene, by the way. What combination? And in whom? And how consistent and replicable or verifiable or falsifiable are these assertions? These are more than trivial questions. Sometimes it's obvious. Sometimes the effects are so strong nobody doubts the result. Much of the time, maybe most of the time, it's very far from obvious. And it's also not right necessarily to say this has an inherent effect. This has an effect in a context only. And then you have to start dealing with what do you mean by context and how many contexts. Because it will have an effect, and we know empirically, even in this particular case, that there are contexts where it doesn't seem to have an effect in the kinds of statistical, inferential sample data that we deal with. What combinations, what sorts of combinations are going to be relevant to the causal question? Now, within a cell, most of what we do is we think of a gene as a unit, a linear unit along a chromosome, and we think of things in terms of what happens right along the chromosome, and the word for that is cis. We think of things in cis, meaning that they happen along a given chromosome. Gene mapping, like I showed you before with that big diagram, is going along the genome point by point and asking for effects. But this is a schematic of the functional units of a gene, and what really happens to make messenger RNA from a, quote, gene, and by the way, most of us are nervous about even the word gene anymore because DNA functions are not the way they used to be thought, is to get this RNA. We've heard a lot about gene regulation, but I just want to point out that many different proteins, often tens of proteins, along with a complex of polymerase activity proteins, have to bind to DNA sequences somewhere around a gene that is going to make messenger RNA. And they have to interact with DNA sequences because these are proteins. They have to bind to this sequence. They often have to interact with each other. Each of these is coded by a gene which has the same structure. And they often have to bind with each other before you get messenger RNA. That means these things are elsewhere in the genome, and that makes this what I would call a trans effect. Cis effect means you just look along the genome. A trans effect means what happens here doesn't just depend on the relationship of things that are here, It depends on things elsewhere. Because each of these genes, each of these proteins are the product of a gene, and they can vary in the same way that all of this can vary, and these binding sites can vary, and when they vary, that affects the binding affinity, and these proteins vary, that affects their binding affinity with each other. We know this. This is not speculation. Now you can think of this in trans effects, which I mean that you can't just look locally to understand effects. Here you have a bed of cells, and you start out with some cells here. They are expressing a cell surface receptor that will look for a signal in the environment. That means somebody has to have told those cells to express that receptor. Then over here and over here, you've got other cells that are expressing some signal molecule. It could be a hormone, it could be a pheromone, it could be a lot of different things. And this signal molecule is going to drift along one way or another, sometimes far, sometimes not. But it drifts along, and there'll be various signals produced, and they'll assemble in this extracellular space. And around here, these cells detect certain of these molecules, but not all of them. And when they detect those molecules, then they differentiate. And that's basically how differentiation happens. That's why we're not just a homogeneous blob. It's all about signaling, combinatorial signaling, combinations of things at a given time and place. And the action isn't always where we look. So if these are the cells that we're interested in, say insulin-producing cells, the action may be elsewhere in cells elsewhere. We may find a genome hit which may have something to do with secreting this signal when what we're really interested in is in this. And we can very easily, and I think often probably are, misled into looking elsewhere for something where we think the cause is, we're looking in the wrong place. And not only that, these kinds of relationships are embedded in one set of these kinds of signaling transactions that influences another, influences another, influences another. We intercept it where we happen to choose, as subjective humans who don't want to get sick, or as somebody studying ecology of grasshoppers, we choose what to look at, but it's embedded in this kind of layered process. And the beginning of it was 3.5 billion years ago, so there's no end. And a fertilized egg has already got a lot of this in action. So it doesn't start with a fertilized egg. Now, trans effects are mysterious as hell. We're just basically even recognizing some of them, and many of you may not even know about them. But I think they should give us a lot more humility than we tend to have when we try to understand genetic causation. In a nucleus of a cell, the chromosomes are all balled up into a ball of spaghetti. This is color-coded by the different chromosomes that we have. And it's being shown in various ways, still very incomplete data, that the very specific aspects of different chromosomes are very close to each other. And those aspects are aspects where genes are being transcribed, statistically speaking. Well, that sounds fine. Okay, so fine. So the chromosomes, they're not just all out in a line like we see in a diagram. They get together. But how do they find each other? How do these parts find each other? Thousands and thousands of parts are finding each other. And from them, transcripts are being made. And if that cell gets a shock, and we've heard about some conditions, environmental conditions, that has to rearrange. Well, how in the hell does that happen? As far as I'm aware, and I've tried my best to find out if there is anything that's known, this is the way chromosomes are schematically shown in freshman genetics to line up before cell division. Chromosome 1 that you got from your mother, chromosome 1 you got from your father in that cell. Nobody knows how they find each other. These are trans effects. They are things that happen in one place, but they involve communication of some sort. Electromagnetic fields, proteins, who knows? Because it's pervasive. It's not just one example that I'm talking about, which has to do with how genes are used. Here's one that I worked on and unsuccessfully, because nobody has worked on it successfully so far. Olfactory receptors are a family of approximately 1,000 genes. You have 2 copies, one from your mother, one from your father, so you have like 2,000 of these genes. They're variably active or inactive differently in every person. Every olfactory neuron in your cell expresses, and these are in clusters of genes of various lengths, some hundreds, some single genes. They're on almost every chromosome. In a given neuron, olfactory neuron, when it differentiates, it picks one gene from one cluster. It inactivates the other genes in that cluster, and the other gene, the gene in the other cluster, and every other one of the 999 genes on both copies in the rest of the cell. Now, if you can tell me how that happens, you'll get another Nobel Prize, because the first Nobel Prize was for the discovery, basically, that one olfactory neuron is expressed per cell. And that's how you differentiate orders, because if your cells expressed all of them, they'd all always be detecting everything, and you wouldn't be able to tell lemon from banana. So these trans effects are pervasive and very important, but they're not in our basic models. And more curious is that when this is the mouse-human synteny relationship, but where corresponding locations are in the different chromosomes of humans relative to where they are in a mouse. And the point I use just to illustrate the point, the same has been shown in a mouse that I just talked about, even though the chromosomes have been all rearranged. How does that happen? How can we have different arrangements of more or less the same genes, because we and mice are basically the same, except they squeak and we talk, and yet these things are robust to this kind of rearrangement. This is a trans phenomenon because it's communication among the chromosomes in the cell somehow. Now, everyone who took freshman genetics knows that if you have a typical Mendelian situation where you have two alleles at a locus, which is rarely the case, and if you have two doses of, say, the big A allele, you're tall, say. If you have two doses of the little A allele, you're small. If you have two doses of the big A allele, you're sort of in the middle. If there are two genes that are doing the same thing, you can get a distribution of small, sort of medium, sort of tall, large. If you have even three different genes that have the same characteristics, which is a simplistic classroom model whose assumptions are not critical to the point I want to make, then you get what's roughly the normal distribution or the Gaussian distribution of a trait like stature in a population. This has only been known for 100 years. And so it's not new information. But what's new is that in the last 20 years, we've started to be able to document some of the genotypes, the sequence variants that are involved. And the key fact to me is that many different genotypes at many different loci can give you the same phenotype. The predictive power from genotype to phenotype is weak, and I'm not mentioning environment yet. The inferential power from phenotype to genotype is typically weak, except that what gets in the news are the examples where it works. And of course it does work. It works because there's a distribution, a spectrum, because life does not guarantee everything to be one way. Things sometimes work out just great. Mendel intentionally chose things that worked out just great, and look what it did for him. So we get a stature distribution. This is a classic figure. Now, if you have evolution, even if you have adaptive selection, what you have is shifting the relative frequencies of these different genes, and on the way, over time, you'll have new variants in different genes arising, and other ones drifting away, drifting out of the population, or being removed by natural selection. So this general picture doesn't depend on non-selection. It doesn't depend on some particular trait. Here's stature. Now, when it's been mapped so far, we've had about 400, I think, nowadays. With large samples, we've had approximately 200, by now, about 200 genes in these kinds of mapping results shown to be contributing to stature, and it's been estimated by extrapolation of allelic effect distributions and other kinds of distributions like that, that if we had a sample of a mere 487,000, which NIH is starting to think is the minimal sample for a real study, if we had a mere, and stature is very easy to measure, we would be identifying something like 700 contributing genes. As is said earlier, mostly trivial amounts. And that would still only explain about 15% of the estimated genetic contribution to stature. So the rest is very, 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 very small effects or something. People have tried to do some hand waving to try to say that this is not what's really going on and it's not so complicated. But I think most of us realize everybody who's the same height has a different genotype. Now, how often do they share things that are relevant to know about is a good question, and there's no one answer to that. But this is not something to do with stature. This is general. We find it in disease. You find it in behavior. You find it in humans. You find it in mice. You find it in plants. You find it in yeast, which are only single-celled organisms. So these are kind of generic findings. Why? Because evolution is not working at the gene level. It's working at the trait level. So a little humility, I think, is in order because we're so far from, when and where do we have to worry about identifying the genes for a given trait when we have this kind of a thing to look for, to face. And one way to view this is that over time you have new mutations arising. They may or may not change in frequency by chance or aided by selection. Mostly they'll disappear. In any given sample, in any given population, or over time, you'll find a pie chart. You can find a distribution of allelic effects some of which are strong enough to detect with statistical significance. Again, by the way, that's a subjective criterion that we choose. 5%, I won't even go into the where 5% came from, but it had to do with somebody tasting whether milk was put into a teacup first and then the tea, or the other way around. That's true. I mean, that's true about its origin. But you'll see that in different samples, you won't necessarily find the same gene or genome region making statistically significant contributions. You don't expect to. There's no reason to expect to, because so many genes are contributing, mostly minor effects. It doesn't really matter which combination you have. And so I would say, even under natural selection, sequence conservation, that is to say comparative analysis and showing that the same gene has similar sequence conservation is a criterion, but it's not a definitive criterion for interpreting mapping results. Because to the extent that something like this is happening, it will not generate sequence conservation, because it doesn't care which genes are making you tall, and even if tall is being good, it doesn't care which genes are doing that. And so replication isn't going to be a good criterion. Nor is falsification, because if you do another study, if you find this gene in this study you don't find it, that doesn't mean this study was wrong. And these are true to an unknowable extent. We don't, because evolution is an ad hoc process. So we don't have a priori theory, like the theory of quantum mechanics and that kind of thing, to tell us, even probabilistically, what we should find. So falsification doesn't work very well. Replication doesn't work very well. Verification is not. Meta-analysis are playing with dynamite, because you don't know whether a meta-analysis should or shouldn't give you a repeatable result. We don't have a priori. The closer the population, the more likely you are to get a result, because populations are connected through a tree of descent. Now, a lot of people have said, well, this mapping looks complex, but when we've had rapid change, like obesity change or other things, stature and so on, that will be not, it will simplify things, because only a few genes are going to respond to whatever it is that caused that to change. But that is just not the way it is generally. Stature is a case in point, so is obesity. They've changed rapidly within a lifetime, as someone earlier mentioned. This is not due to genotype changes. This is due to environmental changes. But it might be that only a few major genotypes are responding to eating whatever it is you're allowed to eat this week. And those are the genes we'll find, and then we'll have the problem will be solved. We don't have to worry about the other genes. Stature is a case where stature is increased like this. And when you look at stature, you do not see a simplification, because it has rapidly increased. Same is true with things like obesity. As a matter of fact, what that means is that we have, did I miss a slide? No. We often, when we look at known functional regions of the genome, we find that they're conserved in their sequence. This is just here's some genes here. The coding regions have darker conservation signal indicators here, which means that if we know a gene in the sense of protein coding, or some other parts of the genome where we know they have real clear-cut function, they tend to be conserved. But if you look at mapping data from massive samples, you don't find things you don't expect to see things conserved, because you see so many genes contributing that you don't think there's not a good reason to think that particular genes are going to be conserved, because selection would not be able to detect, just screening on phenotype, which genotype got you that phenotype. So we have something of an inconsistency. It's a fact, but we have something of an inconsistency, I think, an important evolutionary thing to explain. The general explanation for conservation is that this is due to natural selection. But if these traits are the way I have described, and we've got lots of evidence, natural selection is not going to lead to this kind of conservation. I think actually that developmental, non-competitive selection having to do with gene interaction and environments during, say, embryogenesis will get rid of a lot of gene mutations that would cause problems with genes, and that could be one major explanation for conservation, which is a non-Darwinian explanation, but is still consistent with an evolutionary theory. But it is a problem, I think, that we need to face. When you do mapping, you get these weak results generally. There are always exceptions. And that would not lead you to expect conservation. And yet, when you look for functional regions, you see conservation. This has to do with a major argument over the ENCODE project, which I can talk about in discussion if anybody wants to. I've already just said this. Well, I asked earlier, what about well-posed questions? Is stature, which seems like one of the easiest, most obvious, most cheaply measurable, most clear-cut traits, Is that a well-posed trait to ask what causes it questions, or how did it evolve questions? I'm very much circumspect about evolutionary causal explanations, deterministic or highly specific natural selection explanations, because I think we're far from having adequate understanding of that. But here's stature. You could say, well, stature is so obviously an important trait, but it's not so obvious if you think about it. Stature is the result of a lot of different things, which could have, and probably did have, evolved independently for different reasons, at different times, in different places, with different genomic regions being involved. Net result, well, that's like that. Well, okay, so stature is not such a good measure. Let's take something else. Well, we'll do a cross between two inbred strains of mice. Well, there won't be much variation there, so we'll do a cross, and then we'll, from two supposedly homozygous, strain A, strain B, we'll cross them, and then we intercross them for a lot of generations to generate a mappable database. I won't go into the details, but we've done this for craniofacial measures. And when you look at the distances between measures like the skull width, the skull length, and so on, just between two strains of mice, where at most you have two alleles, and most of the time you have no differences. Each line represent here, going across the mouse genome, represents a different measure and color-coded. And you can see that these peaks and so on basically show the same kind of polymorphism, the same kind of complexity that you get when you look at a natural population. It's scary because it means that it's very hard to tease out things if we're asking well-posed questions. The skull may not be a well-posed, the trait may not be a very well-defined trait. The skull is responding to all sorts of things: language, diets, olfaction, brain, a posture, and so on. Well, okay. Stature, that's easy, but that's complicated, so we'll forget stature. What about obesity? Everybody in here talks about obesity. We hear talk about it all the time. It's a pretty easy measure. We have some ideas about obesity. Here's a map of, this one is body mass index. You go along the genome, you find these various hits, and these hits do not generally, except for one, do not include the experimentally defined genes that are important in obesity, the metabolically known genes, basically. Well, okay, so that's body mass index. There's some genes that have been experimentally studied all over the place and brooded about as major obesity genes or diabetes genes, which should be correlated. They don't, when you do mapping studies, you don't find it. Well, okay, wait a second. What if we know how to advance the slides? What if we do weight to height ratio instead? Maybe body mass index is a crappy measure. So let's do weight to height ratio. Here's your map. There's basically no overlap. Now these things should make us stop in our tracks instead of rushing to nature genetics. They should make us say, what in the hell is going on? When it comes to aging, many things are age-related, and many genetic variants, if they have an age effect, it's a very small effect individually on age of onset patterns, on slope of a Gompertz function or those kinds of things. And those are very hard to tease out, and also they should evolve by genetic drift if they're very late in life and they have small effects. And yet we hear lots and lots of talk about natural selection. Obviously we're adapted, but I think we are far, far from understanding how natural selection works over when it takes like thousands of generations. I think we're way not humble enough in the face of evolution as to what in the heck is going on, because we have these kinds of results that ought to be tractable, and they're turning out not to be very tractable. I said this about age effects. Now I just want to say, nobody argues that the BRCA1 and BRCA2 mutations are a risk factor for breast cancer. I'm not female, fortunately, but if I had one of the known mutations, I'd be getting my mammograms, even though there's a risk of that. But anyway, if depending on the cohort you were born in, your risk of breast cancer by age 60 changes by about double. This is published 11 years ago in Science. It's almost unknown. Nobody knows about this result. If environments, this shows that even with the most clear-cut, strong, Mendel would be pleased to use this kind of a trait. Still, even then, even with the known major mutations, the risk depends hugely on environment. Here's another example from Finland, where the North Karelians had that world's highest documented heart disease rate, and over a 20-year period, they reduced it by 85%, having nothing to do with genes. Well, we all know about genes and environment, but the point is, environments can have massive effects on risk. Obesity is another case. We've heard again about secular trends. Massive, not trivial effects. And there's a worse aspect of that. Oh, here's a tree. Here's a tree, an oak tree from, this isn't from my backyard, but these are from my backyard. These two leaves, this tiny one and this more than a foot long leaf, came from the same tree. Now you tell me how that happens. Well, you say, oh, that is just environment. Well, just environment is a big hand-waving way of explaining a lot of interesting things because the venation pattern is the same. Well, how do these leaves know how to have the same veins, the same lobes and everything when they're massively different in size and they're from the same tree? Well, or are they from the same tree? Maybe it's environment. These are environmental effects, but is it environment? Is this environment? Well, here is the hierarchy, level of the hierarchy is generated by evolution, starting at some point where I didn't want to draw it back because I don't have 3 billion years worth of PowerPoint. We have species differentiation, and species branch off in one way or another. We study animal models because there's variation between species. We study animal models only if we think there's a tractably short evolutionary connection, because then we think things will be shared, which is nothing wrong with that. Then we have populations, and here's different populations, Europe, Asia, Africa, and we do meta-analysis, and basically what we should be explicitly recognizing is that we're most likely to detect are going to be ancient, older mutations that antedate the separation of the populations. To find multiple mutations in the same culpable gene without this kind of analysis, you have to look for rare mutations in families and other tactics. Well, then we have GWAS, which takes individuals from a population, takes their DNA from a blood sample, and then looks for variation in their heart disease trait, or in the case of a tree, in their oak leaves. But what we all have, and while you're sitting listening to me, you've ticked away thousands, if not millions, of mutations in your own bodies, are somatic mutations. We've heard about them in relation to telomeres, methylation, and cancer, and these are potentially very important, and depending on where they happen in development, they'll affect multiple tissues or not. And there's nothing to prevent them being the same kinds of mutations that we look for when we do the supposed inherited genome, and yet every cell is somewhat different. And this could explain why leaves on a tree could be vastly different even if they're on the same tree, because you've got millions and millions of cell divisions on the different branches of the same tree. And I think this is about the end of my spiel, and my point is to say I think there's a big danger of oversimplifying both our ideas of natural selection, our ideas of genetic causation, how deterministic they are, where probability involves causation because of migration, molecules vibrating around and interacting, or where it becomes something we haven't been able to measure accurately. And I think when you think about environments and the other things that have been talked about in this meeting, if you look at them closely, you see we face similar concepts. And I think it's fundamental epistemology, actually, that we haven't really got a good way to understand this kind of causation, which is non-replicable. It's not like Avogadro's number of oxygen molecules in a tank where you figure out the pressure. And I just think right now we're using statistical methods based on that history. They've been hugely powerful for some questions, but we're now getting to the point where we're asking questions for which it may not be very appropriate. Or we'll stick with the things where the effects are strong, tractable, easy. Because everybody's temptation in the room is to say, wow, this is very interesting and all that, but it doesn't apply to me. To me, what I'm doing is fine. Because I'm working with this strong signal. But if you look closely, often you can see that we all face these same kinds of problems. And I think the first talk from Ken Wachter, there are a lot of things about it that I couldn't follow just in a quick talk. But he talked about a single nucleotide variant. He seemed to imply that alleles were words that were used for deleterious variants. And I think there was the idea of mutation accumulation has to be looked at more carefully, I think, to understand exactly what's being said, because when you have context specificity of allelic effects, inherent context specificity, most of the alleles probably have no effect. But we don't know. DNA mutations can make their effect just because of the spacing of the amount of DNA that happens between two places. So you can analyze what they do functionally in all sorts of ways, but unless you just look at the spacing between other functional regions, you may never figure out what they're doing. So I think we have to be very circumspect. my own view, very circumspect about what we're in our own minds thinking DNA causation is like, and therefore what is natural selection doing. When natural selection is pruning weakly at the tails over thousands of generations, it would have very little effect on particular genes. So, but we do see a lot of conservation, and I think we have to try to explain it. And I'll stop. Thank you.
Thank you, Ken.
More deeply.
Thanks very much. Going last at the coffin corner of a meeting when people are tired and some people aren't here is not easy. I'm going to give what will be maybe a kind of rumination or a philosophical overview of some of the things that have been said during the meeting. And since I didn't know exactly what was going to be said, I'll be adjusting what I say about my own slides just because to try to adapt. I think we are faced with some epistemological challenges that derive from our use of what I would say are Enlightenment era, 400-year-old epistemological criteria for science, largely developed for physics. Among the criteria that we use all the time, and many of which have been mentioned here, are replicability, but life, because it's an evolutionary history, doesn't guarantee repeatability, and fitting of survey data, for example, is not the same as prediction unless you have a theoretical basis for doing that. Falsification, you hear it talked about all the time, especially by geneticists. Often, I think, I've never read any philosophy of science, but in the absence of a guarantee of replicability, falsification is not a good inferential criterion. You can falsify a hypothesis because you have a crappy experiment, but you can also harder to make sense of it. So in science, there are things like Platonic idealism, by which the Pythagorean theorem holds for every single triangle, along with the angles summing to 180 degrees, in this case, right triangles, except that we know that there's no such thing as a right triangle, because every triangle is imperfect. And yet, somehow, even on the farthest galaxy, we believe that the Pythagorean theorem would hold. This is a kind of Platonic idealism, that there is some kind of reality to things that we don't actually see except in imperfect manifestations. There are also some criteria for externally derived ways to apply the concept of natural law to the real world. Over here I have Newton's law of, Newton's law of gravitation, which is a deterministic law. If you know the masses of two objects and where they are, you can tell what the attraction will be between them, and you can do it essentially perfectly except for measurement error. In the 19th century, statistical regularity was recognized in things like the ideal gas law, where you know that you can't follow every individual atom, but you treat each atom as a billiard ball, and you say that statistically we can at least predict some aspects like pressure. The implications of ignoring all this are. And what I would say in general is that life is a kind of ad hoc history, which we call evolution. It's a context-specific, and I'll use the word Boolean or combinatorial set of relationships among non-homogeneous polymeric elements, genes, or DNA sequence, regulatory regions, interaction networks. And that's what we're trying to deal with. And a lot of the issues that have come up here, either explicitly or implicitly, reflect this. Often when you have this kind of a setting, a situation, what counts is the collective location or arrangement of these various elements, not their presence or absence. And each trait, each gene, and each population is inherently different. That's what evolution basically means. And life's causal architecture is statistical, not specifically predictable, I think, in principle, just by the way these things act and interact. Now, I would say we should ask ourselves how often we're asking well-posed questions. And this just shows the schematic of a structure of a particular gene, ApoE, that's involved in some human diseases. And what is a well-posed question? Well, here is DNA sequence. It's the ApoE temperature of a volume of gas. These kinds of things lead to externally derived theory that we then apply to new data. And I would argue that in biology, to a great extent, often where it counts for this kind of a meeting, all we have are very inadequate generic theory, and we have to use internal comparisons. Internal comparisons such as in this genome-wide map. If you don't know what a genome-wide map is, this just goes along all of the genome. You look at all sorts of marker variants across the genome, and you decide how statistically unlikely are cases to have more copies of this per capita than controls, and you decide by some subjective and arbitrary cutoff called a p-value or whatever, or likelihood ratio scores or something like that, you decide that this and this point count, these ones don't count, And you're supposed to follow your statistical assumptions, but if they don't count but they're close, you say it's suggestive and you write a grant to follow it up. Instead of saying, I didn't find what I thought and therefore I'm going to go do something else. And then down here, these we say, nah, these don't matter because they don't generate statistical significance. Well, I'll try to talk about that a little later on, about what's a falsify hypothesis, because another sample may not have the same characteristics because we're dealing with life. And life doesn't always replicate because that's what the evolutionary process does. Similarly, verification, you usually want to specify criteria for verifiability of an assertion, but verifiability is a kind of weak criterion because you can't always say how you expect a sample design or an experiment to turn out, for the very reason that life is a complex result of a complex history. Predictability is often said to be the coin of the realm or the gold standard, but even with perfect theory, which we certainly don't have in biology, measurement errors and other things can vitiate predictive power, and things like chaos can mean that even a small amount of error can lead to uncontrolled errors in prediction without our being wrong in any serious way. And finally, I also hear parsimony invoked all the time by people about their explanations, and parsimony is life does not follow a geodesic path. Parsimony is not a guarantee. It's not even easy to define what parsimony would even mean. And life is not parsimonious, so it's easy term to use. It sounds like you know what you're talking about, but it's a lot